So I'm very pleased to introduce our keynote speaker for today's seminar, Catarina Rendeiro, who is a lecturer in nutritional sciences from the University of Birmingham in the UK. And the title of Catarina's seminar today is Food for Thought: Can Plant Flavonoids Modulate Cerebral Vascular Function? Thanks so much, Catarina.
Okay, so first of all, again, thank you for the invitation to be here. It's a pleasure to share this data with the group, especially because my pursuit of the effects on the cerebral vasculature are relatively recent, so it'll be a great opportunity to have your views on it. And thank you so much for the ones that woke up very early for this. So I work in a very kind of niche area of nutrition. I've been mainly interested in how polyphenols and a subset of polyphenols called flavonoids can affect health. And polyphenols are complex molecules you can find pretty much in most fruits and vegetables and plants, particularly flavonoids, which are like just slightly more simpler structures, a supergroup of simpler structures of these molecules, tend to give the very colorful tones to leaves and fruits and vegetables. So it's a good way to kind of know that there's flavonoids in it. It's just to look for color in nature. And in the plants, they tend to have a protective effect. They tend to defend the plant against droughts, against different, sudden changes in temperature, for example. So they do act within, signaling pathways and things like that to sort of protect the plant itself. And when this research started many years ago, the idea that maybe some of these benefits could be transferred by, when we ingest these plants, was kind of the beginning of this. And although you can find most fruits and vegetables will have quite a big mix of flavonoids in it, there are some that are quite enriched in particular classes of them. Okay, and you have here some of those examples, and some of the foods that I kind of worked with throughout my career. So berries, for example, are particularly enriched in anthocyanins that tend to give their very reddish and bluish color. Cocoa, for example, quite rich in flavanols, another type of flavonoid that exists in nature. Citrus fruits only contain flavanols, for example, and so you get the idea. Green tea, again, quite rich in flavanols. Okay, so working with these types of foods in research tends to be a little bit easier because you're kind of dealing with a slightly more specific class of compounds rather than just having a big mixture, because it's usually the issue with them controlling the effects of these compounds. But really, before we go any further, it would be nice to kind of take a few steps back and sort of talk to you a little bit about how the interest in flavonoids, in particular flavanols from cocoa, sort of started, really coming from an observation that was done in this population, the Kuna Indians, which live in a small island off the coast of Panama, and the observation that this population really did not suffer from hypertension as they aged, and pretty much did not have any cardiovascular diseases. And you can see here, that the levels of blood pressure and incidence of hypertension is pretty absent in this population within the island. I mean, the other thing that we people knew is that they had a lot of access to cocoa, to raw cocoa, and so they could have up to 8 cups of raw cocoa a day. But through the years, a lot of the populations in the island migrated to Panama City, and when in Panama City, the intake of cocoa was reduced considerably because they just didn't have the same access as they used to have in the islands. And in reality, what was observed was that the rates of hypertension sort of increased to similar levels to what the Western world sort of experienced. So this was the first link between a food that was rich in these compounds and some protection in the vascular system. And obviously, after that, many studies have been done with cocoa flavanols. I'm just going to give you, a few examples of the initial studies that were published. So, for example, effects on blood pressure, were observed in randomized controlled trials. And this one that I have here was a 2-week intervention in older adults. And I think the interesting thing, if you look at the systolic and diastolic blood pressure here, when these people were on cocoa, either a high cocoa or a low cocoa. And so the high cocoa, so the high flavanol cocoa, is the one in black. And you can see that how days progress, blood pressure kind of goes down. And it's interesting. The same thing kind of happens, with the diastolic blood pressure, a little bit less. And when the intervention kind of stops 2 weeks later, you can see that in a few days, blood pressure kind of goes up again. So this is quite nice cause-effect link that they emerged in the early studies in blood pressure, in terms of, looking at blood pressure. And then, another example of one of the initial studies that showed how flow-mediated dilatation of the brachial artery would change acutely, after one dose of a high flavanol cocoa versus a low flavanol cocoa. And you can see that you have quite a substantial increase that peaks at around 2 hours. This is also around the time that the flavonoid metabolites within the circulation peak. And then after that, we know that they kind of start to get excreted from the body, and the effect sort of goes away in a very kind of mimicking really what happens with the metabolism. Okay, and this data have been replicated many times in young, old. In old populations, in hypertensive, people at risk of cardiovascular disease, etc. Mostly you get the flow-mediated dilatation effects, the FMD effects, both acutely and chronically. The blood pressure tend to only happen, the blood pressure effects tend to only happen in populations that are at a slightly higher risk, hypertensive and chronically, not so much in an acute manner. So one of the main mechanisms that have been sort of put forward to kind of describe how these compounds might be acting in the vasculature has been linked to modulation of nitric oxide. And we had, a few pieces of data that kind of support that. So initially within the Kuna Indians that I just showed you, there was some observational data suggesting that there was considerable differences in the rates of nitric oxide species that were being excreted in urine between the people within the islands and the Panama City. So if you look at the white bars here, which are the urinary nitric oxide species, you can see quite a big drop in there. And that kind of mimics also a drop in the presence of flavanol metabolites within the urine. So epicatechin is just one of the flavanols present in cocoa. And so, I mean, this was a piece of, the first piece of evidence that kind of pointed to nitric oxide. Since then, obviously, in randomized controlled trials, people have shown changes in blood levels of nitric oxide species, after an acute dose of a high flavanol intervention. And it kind of seems to also follow the type of dynamics that we saw with the FMD and the levels of circulatory flavanol metabolites within the blood. And finally, there's been quite a bit of work in cell models that used human coronary artery arterial endothelial cells to look at production of nitric oxide in a dose, you can see here in a dose-dependent manner. So this is like increasing concentrations of epicatechin, the flavanol and cocoa. Apart from looking at nitric oxide, there's been quite a bit of work looking at the signaling that obviously is upstream production of nitric oxide. So, for example, activation of enos, which is the enzyme that produces nitric oxide in endothelial cells, and showing that there seems to be an activation of the main sites of this enzyme, as you can see here, and also even signaling cascades that lead to enos. So, for example, Acute is known to also be activated, and most recently, people identified a potential receptor that is thought to be mediating some of these effects, and that is the G-coupled estrogen receptor here. So it's thought that it is through interacting with this receptor that, for example, epicatechin can trigger this type of responses within the vasculature. And you can see that indeed, if you look at the structure of epicatechin and the structure of estrogen, they're not completely, they're quite similar. So it is likely that, the epicatechin might be interacting at the level of membrane and triggering these types of effects. And so this is just another example of the types of studies that, I've been involved in, looking at the effects of polyphenols or flavonoids in endothelial function. So here we're looking at blueberries, so that contain anthocyanins rather than flavanols, but just to illustrate that, for example, when we did those responses in which we increased the levels of polyphenols, that you have a certain increase in FMD that kind of seems to reach a plateau, and then it doesn't kind of increase further. So there might be a sweet spot in terms of how much we can achieve in terms of benefits for endothelial function. And so, in general, when we talk about flavonoids and vascular function, the idea is that, or what we think that's happening, there's been quite a lot of work within this area, more than, in the brain that we'll get to in a minute, kind of suggesting that these flavonoids might be, regulating, upregulating signaling pathways such as AKT, for example, and activating enos and producing nitric oxide, which leads to a more enhanced vasodilation, which you can measure in things like FMD and leading to eventually lower blood pressure in populations. So in parallel with all of this work that was going on, looking at the effects of polyphenols on vascular function, there was starting to emerge some evidence, and this is like one of the earliest studies that looked at how, what happens in terms of the cognitive trajectory of all the populations that consume higher amounts of flavonoids versus the ones that consume lower amounts of flavonoids. And you can see this here. They follow this 1,640 subjects for 10 years. You can see that the populations that had the higher intake of flavonoids seem to experience a decline in cognitive function that was a lot more attenuated than, for example, the ones that had low flavonoid intake. And so from here it emerged like an interest in understanding, whether these compounds could also be beneficial for cognition. And I did spend a lot of my earlier career actually looking at this, trying to study this mainly in animal models. And obviously the choice of animal models was mainly related to the fact that you can control very well what these animals eat. And we have the advantage of being able to measure aspects of cognition in the animal, but also obtain the brains of those animals and look at some of the changes that might occur within that. And so we've done that in different populations of mice, so young and old. Some of the studies, the studies that we've done, I mean, in most of them we looked at obviously cognitive function and molecular mechanisms, and I'll show you that in a minute. The other advantage that we had is that you can have an intervention that is quite long. So we could give these animals, these compounds for a few months, which is quite a big proportion of their lives. So this first one here, just to give you a couple of examples of some of the experiments that I conducted. So we, for example, looked at what happened, in terms of their memory and learning after you intervene with a blueberry, with a whole blueberry diet. Okay, but then we also gave just the portions of. So in two other different groups, we just gave the different portions of flavonoids that blueberry contains, so the anthocyanins and the flavanols. Okay, so we give it to them in individual to compare to the effect of the whole blueberry. And in fact, what we saw is that the effects seem to be kind of similar if you use just the flavonoids or if you give the whole blueberry together. So that was one of the examples, and we also did some dose response studies in which we show that normally if you do tend to if you do increase the dose, that the benefit that the animal has seems to kind of be slightly higher too. And so this is most of the cognitive studies that we run, and then spending a lot of time looking at the brains of these animals and really try to figure out what was going on inside neuronal cells. And after many years of working on this, we kind of a picture started to emerge that kind of suggested that signaling pathways like Acute and ERK and mTOR were being their activation was enhanced when these animals were in this flavonoid-rich diets, activating very key transcription factors like CREB, which is very, very important for synaptic plasticity within the brain. And obviously key neurotrophins like BDNF was also elevated, as well as some of the subunits of glutamate receptors. So this was kind of, what we're getting to in terms of understanding mechanisms linking cognition, the effects of flavonoids on cognition. Obviously, one of the questions was always, to kind of trigger these types of effects, these compounds would need to cross the blood-brain barrier and actually enter the brain to have this direct effect, similarly to what we've seen in the glial cells when they're in circulation. And although many studies have shown that these compounds can reach the brain, it really remains to be established whether they reach in amounts that would be enough to maybe trigger some of these effects. So I think mostly we believe that they do, but these are not easy things to measure in very tiny amounts of brain tissue. So, but it's also true that as I was exploring this, so these types of modulation of receptor and cell signaling, improvements in neural communication as a consequence of flavonoid intake, which would be in effect a direct effect on brain cells. Obviously, there is another type of mechanism that we started to consider, given all of the evidence that we had of the benefits of these compounds within the circulatory system, and improving endothelial function, and started to think whether, we could actually, some of these effects on the peripheral vasculature could be translated to a certain extent to the cerebral vasculature, and have really result in a more efficient oxygen and nutrient delivery and obviously also contributing to cognitive benefits. Obviously in this case, we do know that these compounds get in the blood, and so you would be looking at an indirect effect, and an effect that can occur quite quickly. Right, within a couple of hours of having a cocoa drink, for example, you do see this effect on flow-mediated dilatation. So, in principle, you could also see it in terms of blood perfusion and cerebral blood flow. So we kind of, after having explored this side of the equation for a while, I started to kind of think about how this could be important also. And so the first study that I did I was very lucky. I was at the University of Illinois at Urbana-Champaign in the US, and I established a collaboration with Dr. Dobrucki, an engineer that had this incredible machine called single photon emission compute tomography for little animals. So we could actually image the brain of these animals and look at their blood-brain perfusion by using a small radioactive tracer that would just go around the body, so across the blood-brain barrier, and be extracted from the blood to the tissue after a single capillary passage. So then we could have a sort of an indication of perfusion. So it's an approximation of, let's say, regional blood flow. The great thing about this was that we could do this more than once in an animal, right? And so for the first time, you can actually measure how was blood perfusion, brain blood perfusion, before we actually start that intervention and after, the intervention itself, which is something that most of the time you don't get to do in animals, because obviously when we extract their brains, we can just do it at the end. We can't look at the beginning. And so we actually went all in and did a 6-month intervention in middle-aged animals. So by the time they finished their intervention, they were quite close to the natural end of their lives. So they were pretty old. And what we observed was that, contrary to the low flavanol group that dropped their global brain perfusion quite substantially over those 6 months, the high flavanol seemed to have preserved blood perfusion more generally. And I put here the individual data of this animal, so you can see that in the low flavanol, it's a lot more consistent. That drop is a lot more consistent. And in the high flavanol, you do have quite big drops, but you also have a lot of animals that seem to even improve a little bit their brain perfusion. The other thing we did, and this was just, again, an attempt to look at whether there would be any differences, regional differences within the brain. So we just used some brain MRI brain atlas to kind of put on top of the scans that we had, and kind of look at some of the main areas of the brain. The reality is that you don't really have with these techniques a lot of resolution. So I'll take this data with a pinch of salt, but in general, what we saw was that it seems like the effect was kind of more or less homogeneous throughout the brain. So our next question was, well, if it seems like flavonoids are having some sort of preserving blood perfusion, is that because they're actually preserving blood vessels, small capillaries within the tissue? And so obviously, because we could obtain the brains of these animals, we decided to pick a region of the brain that is known to be quite vascularized and very important for learning and memory. So all of the cognitive tasks that I've been looking at, in my previous experiments. So we looked at the hippocampus, and we used some immunohistochemistry and stained for collagen 4, and started to count basically blood vessels within this area, and looked at how dense what the density of those blood vessels. And if you look here, if you just look at the whole hippocampus, you can see there's really not any difference between the number of blood vessels that we have there. And then we looked at it within specific subregions of the hippocampus, but again, as a whole, these effects don't really emerge. It doesn't seem like differences in brain perfusion are attributed to preservation of blood vessels. Of course, I mean, all we're doing here is that we're counting blood vessels. We don't know how functional those blood vessels are. And so it might be that, you know, they're there, but they're not doing anything. But certainly we've sort of thought that this effect was probably not a structural one per se, and that we should be looking at measures that are a little bit more functional in itself. And obviously the other thing is, when we're looking at perfusion, you know, we're doing this in a, you know, the animal is anesthetized, is in a, you know, it's resting. So I think the next thing we wanted to do was a little bit different. And so we moved on to humans. Just give me a second. And so this work, the idea behind it was that we wanted to use a functional measure, right? We wanted to challenge the brain somehow. And we decided that doing looking at blood oxygenation in response to a CO2 challenge to hypercapnia would be a good one to start with. Mainly because although the evidence is not completely established, there is some suggestions that a nitric oxide might be involved in this response, and certainly other interventions that manipulate nitric oxide availability, like, for example, L-arginine, have been shown to improve CO2 reactivity. So we thought this would be a good thing to kind of you know, test flavonoids on, and we used functional NIRS to do this. With a bunch of different sources and detectors across the frontal areas of the brain, and you can see, you know, our montage here, and we use this special helmets to kind of put detectors and sources all across this area. I mean, this was a good thing to do because most of the studies that are out there that have done something with NIRS in this area have only used a location. So here we had a little bit more of a robust coverage of this area of the brain. And we went, you know, we just wanted to do something simple, you know, an acute study. So just looking at what happens before and after 2 hours of a cocoa intervention. So we know that that is enough to trigger a positive effect on FMD. And so we just did it side by side, you know, challenge the brain before, challenge the brain after with CO2. And we did this. So the other important thing is that we decided to do this in young, healthy volunteers. So we're actually looking at the physiology in their intact form. Okay, so everything should be working as it's supposed to. So we went for a high flavanol cocoa versus low flavanol cocoa, and really here, the only thing I wanted to say is that obviously we matched everything with those cocoa powders that were not polyphenols, that were not flavonoids. So, you know, the effect that we might see is definitely due to the content in flavonoids in it. And it's quite important that all of these things are matched because, you know, if you don't, then you can't really attribute the effect to the flavonoid content. And sometimes this is one of the limitations of the studies in this area. So the first thing, we just confirmed that the effect on flow-mediated dilatation was there after 2 hours. So everything was working as we expected to. And then in terms of the functional response to CO2, so you have here, so you can see here the whole dynamics of both low and high flavanol. So low in blue and high flavanol in red. And the ones that we really want to kind of focus on is the non-dotted line, right? So the high flavanol is this one here, right, in red, and then the low flavanol after 2 hours is this other one, blue here. And I mean, it's quite obvious that, you know, during that challenge, that it seems like the increase in levels of oxygenation within the frontal areas of the brain is a lot higher, substantially higher for the high flavanol Okay, and this is just a quantification of this difference here. So this was quite interesting to observe that the amplitude of that response was higher for high flavanol. But it seemed to be also a little bit faster. Okay, so if you look, this is like the minutes during the challenge, the CO2 challenge. And if you, for example, compare the low flavanol after 2 hours with the high flavanol after 2 hours, you can see that the effect in oxygenation emerges earlier. So we quantified this, and it emerged about a minute earlier within the high flavanol compared to the low flavanol, meaning that you're not only looking at a higher level of oxygenation, but it seems to also be a little bit faster. And this is just the quantification of time to reach 90% oxygenation within the brain before and after the 2 hours. And you can see that there was quite a substantial decline in time to achieve that level of oxygenation. All the data that I'm showing you here is an average across all of those different points in the brain, mainly because that there didn't seem to be that much of a difference, regional differences, and so we just average them all. So in a way, this kind of mimics, this kind of supports what we've seen previously with the animal data, although obviously it's quite a different, quite different models. In terms of the levels of deoxygenation, those were very similar, so there were no significant differences there. Now, the other thing we've also decided to do was to have a cognitive task at the end of this, but we didn't really look at levels of oxygenation during the performance of the cognitive task. The cognitive effects of flavonoids, there's a lot of mixed data out there. Sometimes for the same task, you have people reporting positive effects and people reporting no effects at all. And sometimes, you have some physiological benefits, within the brain that then don't translate into cognitive benefits. And so we start to think whether, and this is something that has been talked about, previously, whether obviously the level of difficulty of the task, it's key when you're doing these things. And so what we decided was to use a Stroop task, which has been used, many times before, also in this area, but just use another version of it, which has also been published previously, which we call double Stroop. That is just like a slightly more challenging one. Okay, so this would be what you normally would do with the Stroop task. So just match the color that it's here. So in this case, red. But we just made it a little bit more difficult by giving a color to the actual choices. And what that does is that it makes the volunteers take just a little bit more time to kind of work out what they have to do. And indeed, that's what we've shown here. The efficiency really declines, or here you have the inverse efficiency score. So it increased. So from the Stroop to the double Stroop, really showing that, the task was a little bit more difficult. But the interesting thing was that although we did not see any differences in the Stroop task, the benefit of the flavanol intervention emerges within this more difficult cognitive challenge. Okay, and this is just another way of sort of showing that benefit. So this was interesting. I mean, it just gives us an idea that it does matter how challenging it is for the population that you're working with. And I guess this is probably not news, but it might be a reason why a lot of the data out there is a little bit mixed in terms of these effects. And obviously, this is a young population. There's no cognitive impairment to start with. They're perfectly capable of performing these tasks. Now, one last observation that we've done, and this was obviously not something we planned for, but we thought was quite an interesting finding within itself, was that we realized that within our data, we had two distinctive groups of volunteers. Okay, most of them were within group 1, okay, in which, we saw that kind of change in that difference, let's say, in oxygenation responses during hypercapnia between the high and the low flavanol. Okay, so this is more or less what I showed you with the whole data, right? So the high flavanol going up a lot more, higher and a little bit faster. But then we also noticed that there was a smaller group in which there was no difference at all, right, between the two of them, and in which the response was quite high, right? So kind of ceiling type of effect, I'd imagine, okay? Which really is quite, I mean, I think especially in this area that I worked with, we do know that there is a lot of individual differences in the way that people respond. And this is also the case for FMD. And I guess this is what we see here. But it was interesting to realize that, were the ones that already kind of having an oxygenation response to CO2 that was quite high that did not benefit, right? While the other ones, also perfectly healthy volunteers, they did not have such a good response that benefited, okay? So, whether this is driven by levels of fitness of this, of these two groups, we don't know. That was not something we measured, certainly something we will be looking at in the future, because really trying to understand who really benefits from this, even in a young, healthy population, there's those differences are there. I mean, the second thing that we observed that really kind of stunned us to an extent, it was only this, the group that had benefits in levels of oxygenation that also had benefits in the cognitive task. The other ones didn't at all. So that kind of, also, by chance sort of got us to link these two things, which obviously we know are related, which, responses, oxygenation of efficient oxygenation of the brain and cognitive function, that this might indeed be linked, and the ones that really benefit in terms of levels of oxygenation might also benefit in terms of their cognitive performance. And so just to put this in the context of what was there in the literature previously, there's only a few, handful of studies that have been done with flavonoids looking at, physiological measures within the brain, both acute and short term. Most of them have been, people have been measuring flow and perfusion in a resting state, which, might be less informative. And again, there we have some contradictory results. So some people reporting increases after intervention, after others reporting decreases. So only a few that are highlighted here looked at brain vasculature in response to a stimuli, either a cognitive task, or, for example, one study looked at in response to exercise after 7 days of cocoa. And they also, like us, showed an increase in prefrontal cortex oxygenation levels. Another study, Acute study, actually showed increases in blood deoxygenation in prefrontal cortex in the context of normoxia and hypoxia. So again, some variation there, but not a lot of studies. But in general, there's been also some people reporting increases, some people reporting declines. And so we did a study that, we're still working on and hasn't been published yet, in which we decided to actually look at CO2 reactivity in a middle cerebral artery, so like most people would do when looking at CO2 reactivity. And actually what we saw here was that the high flavanol diet seemed to, or high flavanol intervention, so this is again healthy volunteers, exactly the same design, looking at a young healthy population, but we kind of saw like a decline in reactivity after the high flavanol versus the low flavanol. And here you just have that in absolute change. So quite a pronounced decline after high flavanol compared to low flavanol. Obviously here we didn't yet look at the dynamics of how this happened. We're just, reporting that, reactivity, looking at those last, 30 seconds like you would normally do. And this was quite, I guess, surprising. I mean, I'm kind of new to the area. So, one thing that I thought, is it possible that, the micro and the macrovasculature might be responding differently to it, and whether is there an evidence out there, that the vasculature can respond differently, for example, to exercise, depending on what you're measuring. But I would love to hear what your thoughts on it are, or whether you think this is more related to the technique itself. It's more of a technical thing, in terms of the way that we're measuring it. And I also wonder whether it would be interesting to look at, what happens a little bit more upstream, looking at, for example, the internal carotid artery, doing a CO2 challenge, although I have concerns in terms of how feasible this is. It's not easy to be imaging this artery while breathing CO2, but I'll leave it out there. Maybe, some of you might have some ideas. Okay, and so quick summary to close. So obviously, we started off with, just talking a little bit about how flavonoids protect against vascular disease, and the evidence that is out there, kind of linking it to nitric oxide signaling, and also, that. In parallel with this, the evidence coming from epidemiological studies, but also some randomized controlled trials and animal studies suggesting benefits in cognitive function, but whether that occurs through a direct modulation of neuronal signaling in the brain, or some indirect modulation of cerebral blood flow. It's still to be established, probably a combination of both. And obviously, a lot of my, the animal work that I've done kind of suggests that there is a preservation of blood perfusion in aging, and that doesn't seem to be related to density of blood vessels, but we're thinking that it's probably just getting those blood vessels to work more efficiently, at least based on the recent data with young healthy adults related to improvements in brain oxygenation. The other take-home message is that it might be quite important to think about how we're designing our cognitive tasks, whether they're appropriate for the population that we're looking at. And also really this idea of the individual variability in response to diets like this, and what kind of drives why a certain person that seems as healthy as me might benefit more from these compounds than others. Okay. And finally, just this point about the vasculature. When we're looking across the vasculature, shall we expect differences in the benefits if you're looking at micro versus macro vasculature, for example? And so those would be, some of the general ideas. And finally, just wanted to leave like one note to say that obviously, when you think about effects of diet in general, and obviously polyphenols specifically, in terms of, cognitive function, is just that, the place that diet will probably occupy is in terms of sort of delaying a little bit the decline in cognitive, the decline on quality of life that might occur towards the end of life. And so it's just sort of kind of getting this curve to just go a little bit slower. But yeah, I think that's it. I just wanted to thank my collaborators and my research group and my funding, and you for listening. Thank you.
Thanks so much, Catarina. That was an excellent presentation, and certainly on a very unique area of study. So we appreciate that. We do have a couple of questions here in the chat. One from Patrice Prasad. Do we have data about the impact of cocoa flavanols on cerebral autoregulation? So in response to acute blood pressure reductions, for example. And in a related question, is there an increased risk of orthostatic hypotension with flavanols?
I don't, as far as I'm aware, I don't think I've seen any studies in cerebral autoregulation. But I don't know. Yeah, I don't think this experiments with sit-to-stand have been done. Okay. I think it's, yeah. I think only now some of the physiology people like you are, starting to become a little bit more interested in some of this. So I think some of those studies are starting to emerge now, but not that I'm aware of, no.
Okay. So a wide area for study, I guess. So,
yeah.
Excellent.
Yeah, please do.
Do you know, just in a related question, do you know anything about baroreflex function, if that changes with this type of diet?
I don't think that has been looked at also.
Okay.
No.
Okay.
No.
Well, that's good news for people trying to investigate some new areas. Yeah. So excellent. Another question from Mamudreza Tagizadeh. Thanks for the presentation. According to your knowledge, is it possible to delay the decline in cerebral blood flow during exercise by using supplementation of flavonoids before intense exercise?
There have been some studies that combined exercise with cocoa interventions. Now, if I remember well, I think the exercise effect sort of completely sort of takes away the effect of flavanols, because I think it's such a strong stimuli that it, you don't really see an improvement above what you do with exercise. And I think people have done that during exercise and straight after, looking at cerebral oxygenation also in the brain. I can't remember now what kind of exercise, but some sort. But I think in general, yeah, if you have them separately, you have the effects, and then you put them together, and it's just the same as individual.
Okay. And I had a question also about the CO2 reactivity data you presented. Is that using just a traditional 5-minute CO2 stimulus?
Yeah.
Okay.
4 or 5 minutes. Yeah.
Yeah. Have you thought about some of the newer approaches where they're using just a short little burst of CO2 that Ryan Hoyland's really been advocating? So focusing perhaps more on endothelial responses.
No, we haven't, but I guess that could be. So it's just like shorter, and then we'll see what happens. Okay.
Yeah.
I'll look into it. No, it's not something we've considered. So we just did the traditional protocol. 4 or 5 minutes, and then
I think one of the benefits of that approach also is that it's being a short amount or short dose of CO2, being able to measure the ICA flows is a little more straightforward, because you don't have that effect of breathing the CO2 on the ICA dynamics. Yeah,
I see. Okay.
And I have just, I don't think there's any more questions in the chat, but one more question. What are your recommendations for all of us in terms of our diets, in terms of the potential positive effects of flavonoids?
I think really just going, perhaps when you're choosing your fruit and veg, your 5 a day or whatever, that you go for fruits and vegetables that are enriched in these types of compounds. So, maybe choosing apples versus melon, or, I don't know, berries versus, can't think of another fruit without flavonoids in it. But I think it's more in that sense. I mean, and the other thing I would like to say is that when we talk about cocoa, these cocoa powders that we used are treated in a way to preserve the flavonoids in it. A lot of the chocolate that is out there, is highly, unfortunately, chocolate is processed. So that there's not much flavonoids in them, so that they're not a very good source. But you can get them from apples, from grapes, from tea. So from a lot of those other things. I mean, in terms of cognitive function, I just saw that somebody asked which dose of flavonoids are recommended for better beneficial effects of the brain. The latest meta-analysis that was done, taking into consideration all of the other systematic reviews and meta-analysis that have been done, looked at about more than 500 milligrams a day, seem to be beneficial. But this is very variable across foods and even where the foods are grown and things like that. So it's difficult to know, but if you have 5 a day of high flavanol fruits and veggies, you probably are over that line.
Okay.
Yep.
Excellent. Well, once again, thanks so much for your presentation today. I'm going to pass over the chair role to you to introduce your speakers. So,
okay, so let me just presentation here. So we're going to have first Dr. Hayato Tsukamoto. Hopefully I said that right. So he's an assistant professor in the Faculty of Sport and Health Science at Ritsumeikan University in Japan. And he's going to talk to us about cerebral blood flow regulation following breakfast.
I'm Hayato Tsukamoto, and I'm working at the Faculty of Sports and Health Science at Byakusa campus of Ritsumeikan University as an assistant professor. Our faculty is located in Shiga Prefecture, close to Kyoto, and we celebrated 10th year anniversary last year. Before starting my presentation, I'd like to say thank you to Catarina and Cerebral Auto-Regulation Research Network for the opportunity to speak in this interesting seminar. Today, I'm going to talk about the impact of breakfast consumption on cerebrovascular brain health. First of all, I'd like to mention my research field and our faculty at Ritsumeikan. Fortunately, I live in Shiga Prefecture, which has the biggest lake fever in Japan, so I can run every evening around the lake fever in order to maintain my health. Exercise is capable of maintaining and improving health, unlike medicine. Actually, Japanese Ministry of Health, Labor, and Welfare proposed in Health Japan 51 that to maintain health, first, regular exercise should be performed. Second, dietary habit, necessary stop smoking, and finally, we should take a medicine. Accordingly, my research area is exercise, nutrition, and environmental physiology for brain-related health. In our research, we can not only use TCD, but also assess the brain activity using EEG, fMRI, and molecular response such as blood analysis. The theme of this seminar is nutrition and cerebral blood flow. So I'd like to discuss about the impact of dietary habit on cerebrovascular health as a modifiable risk factor. This slide indicates the role of 5 major nutrients. Among them, carbohydrate, fat, and protein are used as energy substrate for each tissue. Of course, brain also constantly seeks energy substrate, and our research hypothesis that cerebral energy metabolism plays an important role in cerebrovascular and cognitive health. I am interested in impact of both macro and micronutrition on the brain, such as flavanol. Today, I'll talk about macronutrient study. So in case of meat consumption, the timing may be important consideration for healthcare. In general, some researchers believe that the breakfast is the most important meal of the day. It is well known that breakfast skipping causes metabolic dysfunction, thereby increasing the risk of diabetes. In addition, as well as peripheral physiology, the negative impact of breakfast skipping on brain as cognitive function in the morning may be observed. Given that breakfast skipping evokes post-lunch hyperglycemia as second meal impact, it is possible that breakfast skipping negatively affects physiological response in the whole day. Nevertheless, up to 35% of young people in many Western countries regularly skip breakfast. So we examine whether breakfast skipping negatively affects cerebral blood flow regulation along with cognitive decline. We assessed dynamic cerebral alteration of middle cerebral artery blood flow velocities following breakfast consumption or breakfast skipping. Dynamic cerebral alteration is an important cerebral vascular mechanism to maintain relatively constant cerebral blood flow against very low frequency or low frequency fluctuation in perfusion pressure for brain homeostasis. Dynamic cerebral alteration can be evaluated by using transfer function analysis. The transfer function phase and gain shift reflect the time relationship and relative amplitude between changes in arterial pressure and cerebral blood flow in the very low and low frequency range, indicating that large phase or decreased gain interpreted as the improved dynamic cerebral alteration. Recently, we found that breakfast skipping, while quite broad, doesn't affect dynamic cerebral alteration, while transfer function gain is improved 2 hours after breakfast consumption here. Thus, breakfast consumption causes better dynamic cerebral alteration in the mid to late morning than skipping. Particularly, previous study reported that the risk of cerebrovascular events may be increased in the mid to late morning. This graph indicates the number of patients with stroke. According to this data, stroke is frequently occurred from 9 to 11 a.m. This time reflects postprandial breakfast. So probably efficient dynamic cerebral alteration is necessary in the morning to prevent stroke, and breakfast is an important consideration for optimizing cerebrovascular health. Moreover, the other study reported that an impairment of cerebrovascular function is observed in the morning, such as dynamic cerebral alteration and CO2 reactivity. Given this, breakfast skipping may increase the risk of cerebrovascular events in the morning when compared with breakfast consumption. Indeed, the Japan Public Health Center demonstrated that breakfast skipping frequency is associated with the number of cerebrovascular disease incidents as well as cardiovascular disease. As you can see here, all p-value of cardiovascular disease, stroke, and cerebral hemorrhage indicate significant main effect for breakfast frequency. Therefore, breakfast should be consumed in order to cerebrovascular healthcare. Until here, I propose the importance of breakfast consumption on cerebrovascular function. From this slide, I talk about breakfast quality. I mean, what kind of breakfast should be consumed, focusing on glucose. It is known that blood glucose, lactate, ketone bodies, which are derived from fatty acid oxidation, are uptaken into the brain. Accordingly, meal consumption actively improve or maintain brain function. Particularly, among them, glucose is known as an essential energy substrate for the brain. Thus, it is possible that postprandial hyperglycemia, which indicates the plenty of major energy substrate, has positive impact on the brain. Probably this hypothesis, answer to this hypothesis is no. We do believe, but it is well known that postprandial hyperglycemia and hyperlipidemia increase the risk of cardiovascular events. Probably postprandial hyperglycemia and hyperlipidemia increase the free radical, which affect vascular function. If so, it is also possible that brain is damaged by postprandial hyperglycemia and lipidemia. For example, as mentioned in the first slide, exercise works as medicine for the brain. This right figure indicates the relationship between brain glucose uptake and the blood lactate as an index of exercise intensity. In general, higher intensity exercise elevates blood lactate level. As you can see here, brain glucose uptake decreases when higher intensity exercise, performed. It is also well known that higher intensity exercise has beneficial impact on brain function as cognitive function and neurovascular coupling. So probably brain glucose uptake and its metabolism do not need for exercise in this brain-related health. Interestingly, blood glucose elevation reduces cerebral oxygenation. Previous study demonstrated that cerebral metabolic rate for oxygen, CMRO2, in response to glucose ingestion. They found that CMRO2 is gradually decreased after 50-gram glucose ingestion, indicating that probably postprandial hyperglycemia attenuates cerebral metabolism, whereas glucose is an essential energy substrate for the brain. In case of milk consumption, glycemic index, GI, should be considered to control postprandial blood glucose. GI is an indicator of how much blood glucose level rises after a meal consumption. For example, compared to Japanese soba noodle, white bread compared to Japanese soba noodle, white bread is higher GI meal, and thus postprandial hyperglycemia should be observed because it doesn't almost contain dietary fiber. In other words, even though the same amount of carbohydrates, postprandial hyperglycemia is caused if the GI is higher, containing dietary fiber is lower. As the same way as the glucose ingestion, the negative impact of high GI meal consumption on the brain is confirmed. However, in case of high GI meal consumption, brain function diminishes after postprandial hyperglycemia. Then, postprandial blood glucose elevation should be returned to fasting level 2 hours after high GI meal consumption. Although blood glucose is already returned, cognitive function is diminished 2 hours after high GI meal consumption. In other words, during postprandial hyperglycemia, cognitive function is not altered, but it is diminished after postprandial hyperglycemia. The mechanism of this phenomenon may be still unclear. Recently, we found that the dynamic cerebral autoregulation is impaired 2 hours after high GI meal consumption. Dynamic cerebral autoregulation was also evaluated by using transfer function analysis. 2 hours after high GI meal consumption, black plot, the phase shift in the very low frequency is smaller than that after low GI wave plot, indicating that dynamic cerebral autoregulation is attenuated after postprandial hyperglycemia. Potentially, it is possible that cognitive decline 2 hours after high GI consumption may be associated with dynamic cerebral blood flow regulation. In terms of chronic diet manipulation, there is evidence from Italy indicating that habit of high GI meal consumption may increase the risk of stroke. One group in this table indicates people who regular low GI meal consumption. And 5 groups in this table indicated people who regularly high GI consumption. As you can see here, habit of high GI meal consumption increase hazard ratio of stroke and ischemic stroke compared to low GI meal consumption. Taken together, high GI meal acutely impairs cognitive function and dynamic cerebral blood flow regulation. Probably the single meal impact is repeated, and thus the risk of cerebrovascular events may be gradually increased. In summary, I propose that post-breakfast hyperglycemia acutely impairs cerebral blood flow regulation, even though glucose is energy substrate for the brain. Moreover, hyperglycemia may be associated with the cerebrovascular health. In terms of timing, breakfast is particularly important consideration for optimizing cerebrovascular health. Thank you very much for your kind attention. finally, I'd like to acknowledge collaborated, particularly Professor Shigehiko Ogo, Exercise Physiological Laboratory, Toyo University, Professor Takeshi Hashimoto, and Professor Damian Berry. Thank you.
Thank you very much, Hayato. A great presentation. Okay, so we have a question here from Pat. So thank you for the interesting talk. I have a question regarding the cerebral autoregulation part of your talk. Considering the poor signal to noise ratio and poor reproducibility of spontaneous TFA versus TFA performed on driven oscillations and the lack of association between different TFA metrics, are you confident to obtain comparable results with a different approach? Multimetric approach using forced BP oscillations such as sit to stand and repeated squat stands, for example.
Yeah, thank you for the question. So that point is very important. So we understand the limitation of this study. So we need to examine the dynamic cerebral autoregulation during, for example, squat challenge, very low frequencies, very low frequency squat challenge, and the confidence should be increased. And we need to examine TFA using point estimated analysis. But as we also think, in general, we need to, we have normal fluctuation of cerebral blood flow, right? So we think in terms of TFA, coherence is decreased in this data, but we also think normal spontaneous fluctuation against spontaneous fluctuation of cerebral blood flow against perfusion pressure is also important. Yeah, so that's why, yeah, I think, yeah, we understand the limitation, but we believe this data is also important.
Thank you. Pat, do you want to add anything else? Or are you okay? Okay, I'll leave it there then. So Carolyn asks, so thanks for the interesting presentation. Do you know the effect of breakfast on bioreflex function and the interaction with cerebral autoregulation?
Yeah, that is also an important point, but yeah, you know, my supervisor is a specialist for bioreflex function. So that's why we need to check the bioreflex function, but not yet. Yeah, thank you very much.
And Pat is also asking, did you measure PET CO2 during your studies? Any difference between baseline and following breakfast?
Yeah, we checked the PET CO2. We confirmed the significant change in the PET CO2. PET CO2 is increased after skipping breakfast, but effect size is very small. So we think this doesn't matter. So yeah.
Okay. And then Binu Thomas asks, thank you for the nice talk. So does the CMO2 drop after glucose consumption show an alternate source of energy provided by glucose compared to energy from O2 metabolism?
We need to check CMRO2 after consumption of rogiamine consumption, but now I don't know I don't know the answer of this question.
Okay, you can always catch up afterwards if you want to continue the discussion. I just have a small question. So in the paper in which you showed the low GI to improve cerebral autoregulation, did you do any cognitive assessments there?
Oh yeah, but you know, we need a sample size to understand the cognitive function, right? Compared to cerebral blood flow regulation. So we check the cognitive function, but no significant difference due to lack of sample power.
All
so
right,
now we are challenging new study using the fMRI. Then we measure the cognitive function.
Okay, sounds good. We'll wait for those results then. Thank you very much, Hayato. I don't know if there's any other questions, but if not, we could probably move on to Dr. Gabriella Rossetti.
Yeah, thank you.
Thank you, Hayato. Hi, Gabriella. Okay, so we're going to have Dr. Gabriella Rossetti. So she's a research fellow at the Center for Integrative Neuroscience and Neurodynamics at the University of Reading in the UK. And she's going to talk to us about dietary nitrate supplementation and neurovascular function.
So thank you, Catarina. I'm honoured to have been invited by Catarina and the CAR network to speak to you about some of my recent work on dietary nitrates and neurovascular function. So my talk today covers two recent publications that I was involved in, both of which were collaborations with Dr. Masahiro Horiuchi from the Mount Fuji Research Institute in Japan, and also with Dr. Sam Oliver from Bangor University in the UK, where I was working at the time. So first, I want to present to you an empirical research study on the effect of dietary nitrate supplementation on dynamic cerebral autoregulation. And then I'm going to finish by briefly covering some aspects of a perspective article we worked on together. So firstly, to the empirical study. And as I'm sure everyone in this audience is aware, the precise regulation of cerebral blood flow, or CBF, is critical for maintaining a constant supply of nutrients and oxygen to the brain and to protect the brain against ischemic injury, capillary damage, and edema. And CBF is normally well controlled at a roughly constant level over a wide range of perfusion pressures via cerebral autoregulation, where vessel diameter in the cerebral vasculature is inversely proportional to blood pressure. Of course, it must be acknowledged that dynamic changes in CBF do occur, both at rest and in response to blood pressure changes. And rapid recovery of CBF through the regulation of cerebrovascular conductance is termed dynamic cerebral autoregulation, or dynamic CA. So dynamic CA can be assessed in a number of ways, including transfer function analysis already covered by Hayato, but also methods to induce transient hypertension, either by rapid deflation of occluding thigh cuffs or orthostatic stimuli, such as sit-to-stand maneuver, accompanied by ultrasound. And under both conditions, the resulting systemic hypertension causes an abrupt drop in CBF, which is recovered only after only a few seconds through a compensatory increase in cerebrovascular conductance. And this capacity for rapid recovery of CBF is, of course, an important aspect of physiology that can be investigated between individuals and conditions. And a previous study by Masahiro Horiuchi and his colleagues at the Mount Fuji Research Institute used the thigh cuff method to investigate the effect of hypoxia on dynamic CA. They observed that dynamic CA is impaired in hypoxia in a dose-response manner, whereby the lower the fraction of inspired oxygen, the worse dynamic CA was impaired. And the impairment in dynamic CA was characterized by a blunted increase in the cerebrovascular conductance in response to the systemic hypertension, preventing normalization of CBF. But the mechanism by which hypoxia reduces dynamic CA is unclear. The physiological mechanisms responsible for the control of dynamic CA are thought to involve a combination of nitric oxide, or NO, alongside neurogenic and myogenic factors. NO is a potent vasodilator and plays a pivotal role in the regulation of cerebrovascular conductance and consequently CBF. Previous research in humans breathing normal ambient air has shown dynamic CA to be substantially reduced when the enzyme NO synthase is inhibited, for example, with L-NAMA, as in this study by White in 2000. And in oxygen-depleted environments, the L-arginine pathway, which is helped by the enzyme NO synthase, has decreased ability to generate NO. And it's possible that this impairment in the capacity for NO production may contribute to the hypoxia-induced impairment in dynamic CA. But NO can also be generated by the serial reduction of nitrate to nitrite and then nitric oxide, providing this alternative pathway for NO production in hypoxia. And dietary nitrate supplementation has been shown to be effective in increasing NO bioavailability, and also, among other things, to improve MCA velocity in normoxia via a reduction in cerebrovascular resistance. In addition, dietary nitrate has been shown to reverse the hypoxia-induced impairment in peripheral NO-dependent endothelial function assessed by FMD. So combined, this suggests that dietary nitrate may be effective to reverse the hypoxia-induced impairment in dynamic CA. And the aim of this study was to determine whether increasing the bioavailability of NO through dietary nitrate supplementation would recover the hypoxia-induced impairment in dynamic CA. And this was also with the view that this could provide evidence that NO bioavailability played a central role in the mechanism of hypoxia-induced impairment in dynamic CA. So this was a double-blind, counterbalanced crossover study with a minimum 2-week washout period between placebo and nitrate. And we investigated the effects of 4 days nitrate supplementation on dynamic CA assessed by the thigh cuff method in normoxia, and then following 60 minutes of hypoxia at a fraction of inspired oxygen of 13%. And for each condition, the thigh cuff protocol was repeated 2 or 3 times at 5-minute intervals to allow for recovery to the resting value. If the first or second trial's reduction in mean arterial pressure was less than 15 millimeters of mercury, a third trial was performed, and then the average of 2 trials was used for that condition. So right internal carotid artery or ICA measurements were performed 1 to 1.5 centimeters distal from the carotid bifurcation with Doppler ultrasound and a linear transducer. For calculation of the rate of regulation, or ROR, baseline values for mean arterial pressure, ICA blood flow, and cerebrovascular conductance were taken from the mean during the 4 seconds immediately before the thigh cuff release. And the ROR was calculated from the slope of the regression line between ICA conductance, which was flow divided by blood pressure, and the time from 1 to 4 seconds after thigh cuff deflation, and then normalized to the degree of cuff release induced hypertension. And I'm sure as many are aware, in previous work, data from 1 to 3.5 seconds after cuff deflation is used to calculate dynamic CA, but we extended this time to 4 seconds after cuff deflation to ensure that we had beat-by-beat blood flow data for at least 3 cardiac cycles. So to look at the placebo data first, just on its own, the degree of hypertension induced by the thigh cuff release was no different between normoxia and hypoxia. But the hypoxia, which is shown in the grey, induced a significant impairment in the recovery of ICA flow following the thigh cuff release compared to the normoxia, which is shown in the black. And in line with the previous study by Hayato and colleagues, this was due to a blunted increase in cerebrovascular conductance. But to add in the nitrate, so the nitrate is shown in the solid lines compared to the placebo in the dotted lines. Dietary nitrate had no effect on the degree of hypertension compared to placebo in either normoxia or hypoxia. But further, it didn't alter ICA flow or cerebrovascular conductance compared to placebo, either in normoxia or compared to placebo in hypoxia. And this is reflected in our overall measure of dynamic CA. So the rate of regulation was impaired in hypoxia compared to normoxia, regardless of supplementation condition. So dietary nitrate supplementation did not influence dynamic CA in normoxia or hypoxia. And importantly, nitrate supplementation did not recover the hypoxia-induced reduction in dynamic CA. So based on this finding, we conclude that insufficient bioavailability of NO is not the primary mechanistic cause of the hypoxia-induced impairment in dynamic CA. And now just briefly, I want to touch on a perspective article we worked on together recently, which brings together evidence on the role of dietary nitrate supplementation in different aspects of neurovascular function. So although the previous empirical study I've just presented focused on experimentally induced acute hypoxia, as obviously this audience know, dynamic CA is impaired in several clinical conditions, which can also be characterized by hypoxia. This includes stroke, anemia, Alzheimer's disease, and it's implicated alongside reduced cerebral blood flow in the development of cognitive function. NO is the primary regulator of vascular tone throughout the body and the brain, not only in response to changes in blood pressure, but also to ensure adequate supply of nutrients and oxygen to meet metabolic demand for neural activity. And this matching of CBF to neural activity is termed neurovascular coupling, and it's essential to enable sustained cognitive function. So to the best of our knowledge, at least at the time of writing the perspective article, only one study had evaluated the effect of nitrate supplementation on both cerebral function and cognitive function together, cerebral blood flow, and cognitive function together outside of exercise conditions. So this paper by Whiteman reported that a single dose of beetroot juice improved executive function on a cognitive task and altered hemodynamics as assessed by NIRS in young healthy adults. And as such, this shows that the only study to include an assessment of the underlying physiological mechanism combined with cognitive function provides support that nitrate supplementation may improve cognitive function through alterations in hemodynamics. But other research in examining the influence of dietary nitrates on cognition is equivocal. And to summarize this literature really very briefly, the findings from acute and short-term supplementation studies do not consistently support dietary nitrate supplementation. And although evidence from chronic supplementation studies is lacking, the only longer-term supplementation study, which lasted 2 weeks, showed that nitrate improved cognitive function in a population that was known to have poor vascular health, and that was in type 2 diabetes. So current published evidence for cognitive function benefits is limited, with equivocal findings drawn from a mixture of single dose and short-term nitrate supplementation. Additionally, convenient sampling in young healthy adults limits our ability to determine the effectiveness of dietary nitrate in populations that theoretically have the greatest potential to benefit, so those with poor vascular health. I mean, as mentioned on the previous slide, the study investigated cognitive function over a longer period in a relevant clinical population did demonstrate a positive effect. But also, studies are generally limited by their lack of physiological and mechanistically relevant measures. And more broadly, an issue of particular importance for us as cerebral physiologists is to ensure that we use appropriate measures of neurovascular coupling when we're investigating relationships between CBF and cognition. So assessing arterial blood flow during a randomly chosen cognitive task or just visual stimulation isn't sufficient to fully characterize neurovascular coupling for a given population or intervention. I believe we have to work with neuroscientists to develop studies that are hypothesis-driven to investigate neurovascular coupling in particular regions of interest or during tasks targeting particular aspects of cognition. So I want to finish by thanking my collaborators on this work, Dr. Masahiro Horiuchi and Dr. Sam Oliver, and also to thank my current collaborators at the University of Reading, where I work as a research fellow at the Centre for Integrative Neuroscience and Neurodynamics. Thank you to Professor Anastasia Christokou, who's the director of the centre, and also my collaborators from the Institute of Cardiovascular and Metabolic Research and the Hugh Sinclair Unit of Human Nutrition. And finally, of course, thanks to you very much for listening.
Thank you so much, Gabriella. That was an excellent talk. Thank you. Okay, so we have a few questions coming up. So from Pat, so very nice talk. Thank you. A few questions. Why have it chosen the tight cuff release method? This technique for quantifying TCA has worse reproducibility than spontaneous TFA metrics. Probably address that one first.
Yeah, Joe, I'm happy to address that now. So I believe that decision was led by my collaborator. So, but I believe the rationale was that it may be more sensitive to interventions, or it had been shown to change in response to similar interventions. So they wanted to compare it there, and in particular with this study, it was really following on from the previous study as well. So the thigh cuff method had been used to demonstrate the impairment in hypoxia. So we wanted to use the same methodology to see if we could reverse that impairment. So that would be my answer. But as I said, it's yeah, of course, there's a lot of other merits to the other methods as well.
And then second question is, would you see similar results in response to transient increase in blood pressure?
It's a really interesting question, and it's not something I've thought about, so I don't know the answer, but I think it's a really interesting question, something worth looking at for sure. Yeah, really interesting question. I think it's a question close to my heart. So similar to when we look at, hypocapnic conditions. And I think we should really be looking at hypocapnic conditions as well. And we should always be looking at stressing our systems in both directions to fully understand what's going on in the relationships. But yeah, we haven't looked at it, and I'm not quite sure.
Okay, so I have a small little question. So did you have any way of controlling for intake of dietary nitrates during the 4-day intervention? Yeah.
Yes, yeah, we did. So participants were given a list of nitrate-rich foods to avoid during the intervention, and I believe beforehand as well. And we do show a marked difference in nitrate levels between the placebo and the nitrate condition as well.
Okay, so
I didn't show it on the
it.
slides, but we did measure it. Yeah, and we did show. I believe the difference was around 200 micromolars or something in that kind of region. So it's quite a substantial difference in nitrate levels between the two conditions.
Thank you so much, Gabriella. It was a very, very nice talk.
Thank you very much.
Thank you. So we're now going to move on to our last speaker. So that's Kamila Pollin. So Dr. Kamila Pollin is a research scientist at the Washington, D.C. VA Medical Center, with the War-Related Illness and Injury Study Center in Washington, USA. And she's going to talk to us about the impact of dietary sodium on cerebral blood flow regulation in healthy adults.
Great. Thank you. Well, thank you all again for attending today, and thank you for the invitation to speak. So today I'm going to present 2 papers that I did during my dissertation while I was at the University of Delaware, working with Bill Farquhar. And it's important to note that my name at the time was Kamila Migdal. And so today we'll focus on the impact of dietary sodium on cerebral blood flow regulation in healthy young individuals. And so to begin, we know that in America, Americans are eating too much sodium daily. The American Heart Association recommends 1,500 milligrams of sodium for optimal health, where Americans are eating well over this at 3,400 milligrams of sodium on average. Additionally, here's a graphic from the US Department of Agriculture showing average sodium density per 1,000 calories. And what you could appreciate from here is that even meals prepared at home have more sodium density per 1,000 calories than the optimal recommendation from the American Heart Association. And so this is alarming because increased serum sodium levels have been linked to adverse cerebrovascular events. This meta-analysis concluded that those with higher salt intake or sodium intake had 23% increased risks of cerebrovascular events, as highlighted here. Importantly to note, strokes are the 5th leading cause of death in the United States. And so there is some rodent literature looking at how dietary sodium impacts brain blood flow regulation. And so I'm just going to focus on one study here. Here they looked at rodents fed a high and normal salt chow, and they looked at isolated middle cerebral arteries. And on the graph here, we're looking at the diameter change in response to acetylcholine, and the rodents fed a normal salt chow or diet. And you can see the expected result. With increasing acetylcholine, we're seeing increases in the diameter of the middle cerebral artery. When they repeated this and rodents fed a high salt diet, you can see that this response was greatly attenuated. So they concluded that high sodium consumption impairs acetylcholine-induced dilation of the middle cerebral artery, and at least in rodents. And so because of this background information that Americans are eating too much salt, it's been shown to be linked with adverse cerebrovascular events, and because some of this rodent literature. For my dissertation, we posed the question, how does high dietary sodium consumption impact cerebral blood flow regulation in healthy young adults, and specifically looking at cerebrovascular reactivity? So I'll focus on the first paper first, and this was an acute study. So we had an acute study and a chronic study. And I'll start with the acute one. So acute meaning that we gave participants a single meal, a single high versus low sodium meal. So for the low was 138 milligrams of sodium. For the high was 1,495 milligrams of sodium. We added that to a half a cup of tomato soup, as pictured here. There was a 1 week washout and at least a month washout for female participants as we controlled for the menstrual cycle. So for our experimental measures, we use a transcranial Doppler ultrasound to measure the right middle cerebral artery velocity. We had beat-to-beat blood pressure with a phonometer. We measured heart rate and end-tidal carbon dioxide levels. So this was our experimental visit. After instrumentation, participants rested for 10 minutes. We then took a blood draw. We then performed the cerebrovascular reactivity test, and this is a representative tracing. So following 2 minutes baseline, we induced hypercapnia with 8% carbon dioxide. We then allowed participants to recover, and then we induced hypocapnia by having participants mildly hyperventilate. In this protocol, we also assess peripheral vascular function using flow-mediated dilation. And so briefly, we use ultrasound to image the brachial artery. We place a cuff right below the elbow. Following 1 minute of baseline imaging, the cuff is inflated. Following 5 minutes, the cuff is rapidly deflated, and we continue imaging for 3 more minutes. And you could see in the far right image that that rapid deflation causes an increase in blood flow, which then changes the diameter. And we calculate flow-mediated dilation by looking at percent change in diameter. We then gave participants the meal condition, and we waited 60 minutes and repeated everything. So for this, we had 37 participants complete the protocol. They were young, healthy, normal body mass index, all were non-hypertensive at baseline or at screening. So looking at biochemical variables, focusing on the serum sodium, you could see that serum sodium increased both following the low sodium condition and the high sodium condition. However, it did increase to a greater extent following the high sodium meal postprandial and compared to the low sodium meal postprandial. Brachial blood pressures, although there was a main effect of time, meaning that you could see systolic and diastolic blood pressure increase slightly, there was no effect of the meal. So there was no effect of the sodium conditions on blood pressure. So as a reminder, this is our cerebrovascular reactivity protocol, and we separately analyze reactivity to the hypercapnic stimulus and separately to the hypocapnic stimulus by looking at the percent change in middle cerebral artery velocity divided by the absolute difference in carbon dioxide levels. So this is our reactivity to hypercapnia response. Contrary to what we expected, cerebrovascular reactivity to hypercapnia was not lower or impaired following the high sodium meal compared to the low sodium meal. There was no differences. Similarly, when looking at the hypocapnic response, when participants mildly hyperventilate, we also are seeing no differences between the conditions or any time point. We also factored in any potential changes in pressure by looking at conductance. So on the left, we're looking at cerebrovascular conductance to the hypercapnic stimulus, and the right to the hypocapnic stimulus. And similar conclusions here, we saw no differences following the meal conditions. So as a reminder, we did measure flow-mediated dilation, and it was with intention to replicate the findings from this paper published in 2011, where they used similar sodium concentrations, and they found that FMD was reduced 30 minutes and 60 minutes postprandial. And so we repeated our flow-mediated dilation 60 minutes postprandial. And again, as a reminder, we calculate flow-mediated dilation by looking at the percent change in the diameter of the brachial artery. And contrary to what we expected, there's no differences. So FMD was not lower following the high sodium meal. And in the table below, just presenting the baseline artery diameter, the peak shear rate, and the shear rate area under the curve, you could also see that there were no differences. So to conclude the acute protocol, contrary to what we expected, cerebrovascular activity was not impaired following the high sodium meal. We also didn't see any differences in peripheral vascular function using the flow-mediated dilation technique. So some concluding thoughts. Well, we know that there are limitations with transcranial Doppler. Of course, we can't image the diameter of the vessel, so we're using middle cerebral artery velocity as a surrogate of flow. And also, a single high versus low sodium meal might not be enough to elicit any changes in cerebrovascular activity in these healthy young individuals. And so that leads us to our chronic protocol, which was a controlled feeding study. So we'll go through that now. So controlled feeding study, meaning that we placed participants on 3 10-day diets. We gave all the food to them. So the first diet was a low sodium diet of 1 gram of sodium per day. The second was 2.3 grams of sodium per day, the medium sodium diet, and the high was 7 grams of sodium per day. These diets were randomized and separated by at least 4 weeks. On the 9th day, participants collected their urine, and on the 10th day, they came into the laboratory following a fast for their experimental visit. And so the experimental visit and the measures were the same as I showed previously. We used the TCD to assess the right middle cerebral artery, and then we repeated the same cerebrovascular activity protocol. So this study, we only had 10 participants that completed, mean age of 30. Again, they were young, healthy, non-hypertensive at the screening visits. So looking at the biochemical and resting hemodynamic measures, as expected, urinary sodium excretion increased in a stepwise manner. This suggests participants were eating the diets that were provided to them. However, serum sodium levels were not different between the 3 diets. Also, systolic, diastolic, and mean arterial pressure, seen here at the bottom, were not different between the 3 diets. So looking at our cerebrovascular activity response, contrary to what we expected, cerebrovascular activity to hypercapnia was not lowest or impaired following the high salt diet. There were no differences across the 3 diets. Similarly, there were no differences to the hypocapnic response. When looking at conductance, taking into account any changes in pressure, similarly, no differences in cerebrovascular conductance to the hypercapnic or hypocapnic stimulus. So to conclude our chronic protocol, the high salt diet increased urinary sodium excretion. So we actually saw a stepwise increase from the low to the medium to the high, suggesting participants were eating the diets. However, we didn't see any differences in cerebrovascular activity to hypercapnia or to hypocapnia. So contrary to our hypothesis, serum sodium was not different. Some thoughts of why that might be is, well, we know that serum sodium levels are tightly controlled in the body. Additionally, this was a fasted blood sample compared to our acute protocol where we assess serum sodium after they ate the soup. Of course, there are limitations with transcranial Doppler. So some conclusions from the study is, well, maybe the brain is resilient and especially in these healthy young individuals against dietary sodium, but there could be some methodological issues. And we know that transcranial Doppler, we're not imaging the diameter. We don't know what's going on. So repeating some of these protocols with different methods, getting a better assessment of flow, cerebral blood flow is important. So that leads me to my future directions. First, repeating these studies, especially the chronic feeding study in a larger sample size. I know that chronic feeding studies are expensive, so doing maybe thinking about using salt pills as a way to get people to eat or have more sodium in their diets. There are limitations with transcranial Doppler, like I mentioned, and then of course, investigating these dietary sodium manipulations in other populations. So both these studies focus on healthy young individuals. So looking at how salt might affect brain blood flow regulation in older adults or individuals with salt-sensitive hypertension is necessary. And so with that, I would like to thank my mentor, Dr. Bill Farquhar, and my teammates, Dr. Robinson, Dr. Watso, Dr. Babcock, and everyone else that helped with this project while I was at the University of Delaware. Thank you.
Thank you very much, Kamila. That was a great presentation. Very, very interesting. Okay, so we have a couple of questions. I'm just going to ask one first. Did you look at what was the habitual levels of sodium in the populations that you studied before they got into the
Yeah, that's a great question, and one that I get all the time. So we did assess, so we had food logs for the chronic feeding study. So there were only 3-day food logs. So it was not enough to get habitual sodium intake. We definitely, doing this study in the future, would like to get a better assessment of that. But just based on these 3-day diets from these 10 individuals we have, they're eating typical American diets, so eating around the 3,000 milligrams of sodium daily. But yeah, we definitely would like to get a better assessment of habitual salt intake.
So they are already taking high levels of salt. Yeah.
So these were college-age students eating in the dining halls. So, as expected.
Yeah. Okay. Yeah. I'm just thinking whether the interventions in a population that is already kind of have high levels of salt, whether actually, makes any difference. Maybe they're already impaired to some extent. I mean, it might be the case that you don't really see it in a young population, that's it. But we're finding that more and more what people are doing in their daily lives influences how they respond to our dietary interventions. But yeah, that's great. Thank you. Okay, so Caroline says, thank you for a great presentation. Were you surprised by the very small increase in serum sodium following the high salt meal in the acute study? And was there any effect of basal serum sodium concentration on responses to the meal or the cerebral vascular outcome measures?
Yeah, both great questions. So in response to the small increase in serum sodium, not exactly, because like I mentioned, the serum sodium levels are tightly controlled in the body. So we don't expect them to go too high. And so that was more of an expected result. In terms of looking at the basal serum sodium concentration and the responses to or cerebrovascular outcome measures, we didn't examine that, but that's definitely something that I can look at since I have the data, but we didn't specifically look at the basal serum sodium or the baseline serum sodium concentrations and the cerebrovascular outcome, but something I could definitely look into.
Okay, thank you. So, and Pat says, what was the rationale for studying cerebrovascular reactivity in your protocols over other important mechanisms such as neurovascular coupling and cerebral autoregulation?
Yeah, that's a great question. Thank you for that. So the main reason is we knew that dietary sodium impacts peripheral vascular functioning using flow-mediated dilation. Like I presented the acute paper, we've also seen that in the chronic literature. And then there is some suggestion that FMD and cerebrovascular activity might have similar mechanisms. Of course, that's highly debated. But at the time when I first started my dissertation, that was my rationale for why we wanted to look at cerebrovascular reactivity. We did look at autoregulation in the chronic controlled feeding study. However, we only looked at spontaneous changes in blood pressure because of the nature of the study. This was a larger clinical trial, and so we couldn't have them stand up or do sit to stands or squat to stands. But we didn't see any differences in autoregulation, but definitely doing dietary sodium manipulations and other assessments of cerebral blood flow regulation are important. Thank you for that question.
Thank you. So we have one more. Did you monitor fluid intake in the chronic study? Do you think alterations in their fluid intake habits and therefore fluid volume differences could be interacting with your peripheral and cerebral outcomes?
That's a great question. So in the acute protocol, we monitored fluid while they're in the laboratory, so they could have water if they needed after the soup, but then it was matched. In the chronic study, we also monitored water intake during the 10 days diets. They had to record that. So it was a subjective assessment of that. We didn't see any fluid intake differences across the 3 diets, which was kind of unexpected. So they could drink water ad libitum, and you would expect that the high salt diet, they drink more water, but that wasn't the case. So we actually didn't see any fluid intake differences across the 3 diets. So we don't exactly know how the fluid intake habits could impact these results.
Yeah, I guess in a way that was lucky, so you could compare it.
Yeah.
Otherwise you'd have that other factor. Okay, so just one last question from me. So you were to some extent expecting some FMD changes, right? But not necessarily blood pressure changes. Is that right? Or there was some evidence that blood pressure could change acutely?
So we weren't expecting differences in blood pressure changes. These are healthy young individuals. We expected them to be salt resistant.
Okay. All right. Thank you so much. So I don't know if there's any other questions for Kamila. If not, I think that's all from me. Thank you so much for your presentation. It was really, really great to hear this research. Not necessarily know if it's good news or not, but anyway, Thank you. I don't know, Caroline, if you have any last comments before we close?
Yes, I will have. So thank you very much to all the speakers. It was very interesting. I had a lot of questions, so this is always good. So again, thank you very much. And I would like to just share my screen to invite you to our next virtual seminar like this. And it will be, it has been organized by Barky Davenport, who will be the keynote speaker. And it will be about cerebral blood flow and pregnancy. And it will include abstract talks by Brittany Matenchuk, Ain Brisling, Rachel Scow, and Kathleen Miller. So we are looking forward to seeing you there. Thank you very much, and have a great day or night or beginning of morning.
Thank you. Bye-bye.